MIT does a great deal, I think, to foster innovation. First, there's like good role models, and one of the things they have, which has been great, is the undergraduate research opportunity program, which is a terrific program. Almost all undergraduates participate in it. And this program is a terrific way for undergraduates to learn research, and it's part of what makes MIT unique. Secondly, I think MIT goes a couple steps beyond just the education. MIT's always had strong ties to industry. There's a terrific technology transfer office. Most people would consider it the best in the world, and also an industrial liaison program that has ties to hundreds of companies that can visit and that people can interact with if they want. So MIT have all these things that expose students and professors to a very broad spectrum of different types of things. And I think that really encourages innovation very well. Thank you. It's a pleasure to be able to speak with you. I'd like to go over some of the work we've been doing at MIT and with collaborators on drug delivery systems. And first, let me tell you how I got started on this. I was a graduate student at MIT, and I got my doctorate in 1974. And pretty much all my friends went to the oil industry then, because that's what chemical engineers did at the time. And I thought I would do the same. I applied to oil companies, and I actually got 20 job offers, 4 from Exxon alone. But I wasn't very excited about that. And I was looking for a way to use my chemical engineering education to help people. I ended up doing postdoctoral work at Boston Children's Hospital with a surgeon named Judah Folkman. And he had an idea, if we could show the next slide, that basically said that if you could stop blood vessels from growing, that might be a way to stop cancer. And just briefly, if you look at the slide, his idea was that a tumor starts out, as an abnormal cell on the bottom left, then it grows as a 3-dimensional mass. And what happens is it gets, if you go to the middle of the screen, middle lower part of the screen, what you see is the tumor is maybe 1 million cells, 1 millimeter in size, and it runs into a nutrition problem. It can't get nutrients in the center or get rid of waste. So it doesn't get bigger than that. But his theory was that the tumor secreted a chemical signal called tumor angiogenesis factor, TAF. And that causes surrounding blood vessels, which were normally quiescent, to start growing to the tumor. And it would get bigger and bigger and bigger. And it would actually, when you go to the top of the screen, what you see is it actually is very vascularized, and it can spread through those blood vessels. That's a process called metastasis. And that eventually is what often kills people. His theory was that if you could stop the blood vessels from growing, that might stop cancer. And as I'll point out, it will maybe also stop other diseases. Now, one of the key problems in this area was that there was no bioassay. That's ultimately so key when you're trying to do drug discovery on a brand new concept like this. So we had to invent a bioassay. And if we could go to the next slide, the bioassay that we wanted to invent was to take certain tumors that you could put in the eye, say of a rabbit, and those tumors would, like V2 carcinomas, would cause blood vessels to grow from the edge of the cornea to the tumor. But it would take maybe a number of weeks, 8 or 9 weeks, for that to happen. So our idea was that we'd have a little delivery system, which might be a polymer or lipid or something else, that you could insert right next to it. And it would deliver the tumor angiogenic inhibiting molecules. Now, it turns out all those molecules were very large. And so we needed to be able to deliver them from this delivery system and meet 2 criteria. One, it had to come out. And that had never really been done before. Large molecules had never been able to be delivered. Secondly, it should be non-toxic, and almost everything causes a problem for the eye. Now, Dr. Folkman was on the board of the one company in the world, ALZA, that had done any work in this area. And they had like Nobel Prize winners in chemistry on their board, and he asked them if they could help. But what they said, next slide, is that they couldn't, because they said these molecules we wanted to deliver were too large. They couldn't diffuse through solid materials, and any more than we could walk through a brick wall. and any of us. Now, the literature said the same thing. Next slide. The literature said that the use of these polymer matrices was virtually restricted to small molecules. But if we were going to solve this problem of angiogenesis, we had to have a way to do this. So I spent several years in the laboratory experimenting with different materials, different designs. I found over 200 different ways to get this to not work. But eventually I was able to make little microspheres or nanospheres. And what the next slide shows is some pictures of one on the left and cut in half on the right. And like I say, almost everything we did not work, but one type of design did. And the next slide shows that what we could see in a paper that Dr. Folkman and I wrote for the journal Nature. And in this paper, we showed that you could deliver any of these macromolecules, like proteins. Also, we showed for the first time ever that nucleic acids could also be released. And those are things like DNA or RNA. Nonetheless, when I first presented this work at a scientific meeting, and I was 27 years old at the time, and I thought I did okay, but and I thought, you know, the older scientists there would want to encourage me, this young guy, but that was not the case. When I got done, a whole bunch of them came up to me and they said, "We don't believe any of this. We don't believe what you just said." And in particular, what the next slide shows is what they thought. And they said that these large molecules are just too big to diffuse through solid materials. Like I said, just like none of us could walk through a wall. They also said that the solvents you were using, they would destroy peptides, proteins, or nucleic acids. And actually things went downhill from there. My first 9 grants were rejected very badly. People would say that Dr. Langer is a chemical engineer, you know, and he doesn't know anything about biology and even less about oncology. Also, I like being a postdoc, but a lot of my friends told me it's probably not good to be a postdoc forever. So I started applying for positions in chemical engineering departments, my major discipline, and no department in the world would offer me a position. So I ended up getting a job in a nutrition department because Dr. Folkman knew the head of that department. But he offered me a job, but he was kind of like what I'll call the benevolent dictator kind of department head. And he didn't ask anybody else in the department what they thought, which might have still been okay, except for the fact that the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. In fact, the next slide shows what Mike Marletta, a good friend of mine at the time and still, had to say many years later, reflecting back on his time and mine at MIT in the late 70s, 1970s. Here's what he said. One evening, he went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said, a senior scientist said, quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. So that was discouraging, but I just kept trying. And one of the things I mentioned is we had done all this work now on the delivery system. So we could do that bioassay that I mentioned earlier, as shown on the next slide. And what we did is we literally put maybe 100 fractions, different isolates, in all of which were large molecules, in these delivery systems and tested them in this assay. Now, every one except for one did not work. I sometimes think of those as the controls. And let me just show you some pictures on the next slide. And what you see, this is from a paper Dr. Folkman and I wrote in Science in 1976, 45 years ago. And what you see on the left-hand side is if you don't have an inhibitor, this is 8 weeks after the start of the experiment, you get the sheet of blood vessels growing towards the tumor. And if you let this keep happening, eventually it would be three-dimensional, the tumor out of the orbit of the eye. It would kill. We sacrificed the animals before that. But when you put an inhibitor in, notice on the right-hand side, and we repeated this 21 times, the vessels are lower, they're sparser, they avoid the tumor. And 60% of the time, these tumors never took off. So we published this in 1976. It still took another 28 years from this paper before the first blood vessel inhibitor was approved by FDA. But using the techniques that we had developed and literally spending billions of dollars, companies like Genentech were able to do this and get approval. And the next slide shows what happened starting in 2004, which is really striking. One after the next inhibitors approved by FDA. Avastin is maybe the number 2 or 3 best-selling biotech drug in history. And not only that, what you see is you can treat not only many kinds of cancers, but for the first time, you could also stop eye diseases that cause blindness, like macular degeneration or diabetic retinopathy, with drugs like Lucentis and Eylea. Also, one of the other things that we thought about was that the delivery systems might be useful for other molecules. As a proof of principle, Larry Brown, one of my graduate students, put insulin in a little aspirin-sized pellet and showed, if we go to the next slide, that it can lower the blood sugar level for 100 days with a single implant. Now, Dr. Folkman one day said to me, he said, Bob, we should file a patent on this. And now that might seem commonplace today, but in the 1970s, Boston Children's Hospital, one of the best hospitals in the world, they never had a patent. So we filed one for the first time, and 5 years in a row, the examiner rejected it. And so the administrative people of the hospital told me I should quit doing this because we're wasting so much money. But you can probably tell by now I don't like to quit. So I started to think, how could we convince the patent examiner to allow this? And as I told you all in the beginning, everybody said this was impossible. It would never work. I wondered, maybe someone wrote that down. So in 1982, I did what's called the science citation search. I looked back at our Nature paper to see who talked about it. And let me just show you one article I found. And I had no idea it was written, but this was from 1979 by 5 of the top material scientists in the world. And what they wrote describing this field is they said, generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising is a really good word for a patent examiner, some surprising results that clearly demonstrate the opposite. So I showed that to the administrators of the hospital. They flew down to Washington to show it to the patent examiner. And he said, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from each of these 5 people that they really wrote that. So I wrote them, and they were nice enough to write me back they really wrote it. And on the next slide, we got this very broad patent. And with that patent, we got involved in licensing things to companies, teaching different companies, starting companies. And what that would ultimately lead to is on the next slide, many, many products that help hundreds of millions of patients every year who might have heart disease, different types of cancer, brain diseases, and so forth. Now, even though we could do this, and make even tiny particles, when you inject them into the body, sometimes they get taken up by macrophages. And also sometimes they stick together and get bigger. So what we decided to do with Vladimir Torchelin, next slide, is create polyethylene glycol coatings, which might make, it's almost a disguise for these particles. And you could even go further and put a targeting molecule on these if we go to the next slide. Let me actually, because I worry that I always don't explain this perfectly, Nova, the TV show, they actually did a video of this. Let me put that on next slide.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
So you could use this to deliver small molecules, but what's really exciting is that you could even deliver genetic molecules like siRNA or messenger RNA. And the next slide is an example of siRNA. I was on the founding scientific advisory board of Alnylam, and my student, Kenna Kink, worked there. And with Alnylam, basically you have ways to silence RNA, but what you do is you put it in a lipid nanoparticle that targets the liver, and this led to Onpattro, which was approved in 2018. Now, also in 2010, going back to 2010, if I could go to the next slide, what you can do or what we did, myself, Nubar Fayan, Derek Rossi, and Ken Chen started a company called Moderna. And the idea here is that DNA makes RNA makes protein, and protein therapeutics were usually successful. But if you could make messenger RNA, you could do it very quickly and inject it into the body, and it would make a protein. But of course, you have to protect it and get it into cells, and that's where the nanoparticles help, or are essential, I should say. And in fact, you could also do other things with messenger RNA. For example, you could treat intracellular and membrane-bound diseases, as shown on the next slide. But when we did this work, and when we started Moderna, all the way through, lots of scientists and stock analysts would criticize it. In fact, on the next slide, this is the Boston Globe, my home newspaper, in 2020, and in May. And they talked to different experts, and all those experts started saying, this was front page headlines, that what Moderna and I were doing, that's not how you do science. But I think those people are obviously wrong. Let me actually go over what Moderna was able to do. Next slide. So the Chinese published the virus genetic sequence in January 11th last year. 2 days later, the messenger RNA vaccine was finalized. Just about a month later, the batches were shipped to NIH. And by March 16th, people were being tested. And by the end of the year, it was approved for emergency use authorization. And as shown on the next slide, as people probably know, it's been used all over the world for hundreds of millions of people. The Pfizer BioNTech one also uses nanoparticles. So that's been exciting. But I want to also talk about, could we someday even deliver these large molecular weight drugs orally? And if we could go to the next slide, what you see, and I'm sure everybody knows this, that when you try to swallow a pill orally, it's just too big. It can't diffuse through the intestine, and degradation also limits what can happen. So by proteases and so forth. So, but people have injected things into the gut. If we could go to the next slide. Can we put all of it up, please? So people have injected, basically what people have been able to do is inject through your rear end. And when you do that, it's very efficient. You don't get much pain because there's very few nerves in the stomach, and you get rapid tissue regeneration. So so that's good, but of course nobody wants to take their drugs through their rear end. So we came up with a special pill. I'm going to show you this. And we did we're funded and did a lot of collaboration with Novo Nordisk. And this is the pill. Let me actually show you a schematic of it on the next slide. So what we do is we have this casing. You'll see this hole. The hole is here. So that's oriented. That's at the bottom. But now we have a needle, and the needle is made of insulin, because that's the drug that Novo Nordisk was interested in. And you can make it pretty much almost 100% insulin, but you don't want it to be shot out of this pill right away. What we do is we have a spring that can shoot it out, but the spring is actually embedded in a sugar glass. And that fixes it. So nothing can happen until the sugar glass breaks. But when the sugar glass breaks, all of a sudden the spring will release the needle, and the needle gets shot out of that hole right where you see that 5 millimeter thing. But there's one more thing, and you might ask, well, what's the timing? So the timing is you don't want it to be shot out immediately, right? Because you're in the mouth. You want it to be in the stomach. So probably you make sure the timing, and I'll get to this, is maybe 30 minutes to 60 minutes later. But there's one other issue you might be thinking about. How do we know that the 5 millimeter part where you see that is facing the stomach wall? In other words, how do we know that this little thing is facing the stomach wall all the time? So what Gio Traverso and Alex Abramson did is we modeled this after what's called a leopard tortoise. And that will always fall and self-orient itself. And the next slide shows an example of that. It just tumbles, it keeps tumbling, but will always fall on the wall. And the next slide just shows a further example of that. It will always do it just because of the weight distribution. And that's what the tortoise does too. Just to show you, if I could go to the next slide about the spring, you can control the time of actuation by the diameter of the sugar glass. And here you actually see the spring shooting out. It's really instantaneous. So once that happens, the insulin gets delivered. And if we go to the next slide, what you see is perforation is really controlled by the actuation force. And if you look at the histology, which is on the right-hand side, really there's no inflammation, even at 9 newtons force. And it's also, our drug is quite stable. If we go to the next slide. So the soma is more stable than, if we look at purity and things like that. So, and finally, does it work? Well, here we did pigs, and the next slide shows that if you look at this, that you get pretty much identical, statistically indistinguishable uptake of insulin compared to subcutaneous injections when you compare it to taking it orally. And what the right-hand side shows you is the histology, no damage. And not only do you get similar insulin delivery, you get similar glucose lowering. And the next slide shows that. No difference again compared to sub-Q injection. And again, histology on the right is quite normal. So the last thing I want to go over is, could we even deliver cells to people? And on the next slide, I'm going to show you a picture of a little baby who's the patient of Jay Vacanti, who's my collaborator. And Jay and I came up with this idea of how to deliver cells, which is shown on the next slide. And that's largely called tissue engineering today. But the idea is we could take any cell type. You see these on the right, like bone cells or cartilage cells. You could, by the way, take stem cells, but we published this before people even talked about stem cells. This paper, by the way, in Science that we wrote has been cited over 8,000 times. But the idea is that you have this scaffold, you grow the cells on it, and then you could make any of these things like bone, cartilage, liver, intestine, ureters, whatever you want. And the next slide just shows pictures of scanning electron micrographs of the fibers. By the way, the fibers will ultimately dissolve and you'll get pure tissue. And just to give you a real example of this, which we've licensed to companies, is you can make artificial skin for burn victims on this. And let me just show you a little baby who is badly burned on the next slide. And you could take the product, which is neonatal skin fibroblasts, on a scaffold and put it in this child at the time of injury. And that's a picture of the scaffold. The next slide shows what it looks like when you put it on the child. The next slide shows you what he looks like 3 weeks later. He's much better. And the next slide shows what happens 6 months later, and he's pretty much healed. And people are using these approaches, including many of our students, to make new pancreases, new spinal cords, to help people with hearing loss, to make new blood vessels, and many other things. And so these are all exciting and I think very important applications. And finally, you could even make a tissue or organ on a chip. And just to pick one example, let's take heart, which is being done by Gordana Vaniak and Milika Ritzik at a company called Tara Biosystems. And here you see heart muscle cells put on a scaffold, and they make the characteristic structures that you'd actually see histologically, sarcomers and intercalated discs. And if you do a high-powered video of these under a microscope, it's amazing. What you see on the next slide is 6 million heart cells, heart muscle cells. And we're not shaking this or anything. They're just beating like one. And like I say, you can just look at it. They're just beating like a real heart. And if you take, and you can even take an EKG on these, as shown in the final slide. And what, of course, Tara is doing is they're doing drug testing on this. You can test drugs for pharmaceutical companies and greatly minimize the amount of animal testing and perhaps human testing you'll have to do someday. And there's other organs and tissues on a chip too that people are developing. Gio Traverso, who I mentioned, he's developed an intestine on a chip. And we ourselves are working with Leeway Tsai at MIT to make a brain nanochip. So there's, I think, many exciting things that can be done to really mimic what the body does and help in drug testing. So I want to thank you for having me. It's been an honor and a pleasure to speak, and I wish you the best at the rest of the symposium. Thank you so much. So I'd just like to share my personal experience about what I found to be useful in helping innovation in general. And these are things that I always tell my own students, and also, points I sometimes make in different lectures I've given. But first, I think, is to do things that you hope will have a very big impact on the world, as opposed to just incremental research. The second thing is to realize that there's something called conventional wisdom. That may be what people will tell you, including very well-known scientists. It may be what's written in texts and articles. And it's important to realize that it's not always correct. If you look back at the history of science, many times you'll see different discoveries have been made that really challenge conventional wisdom. That's certainly been true of our work on drug delivery, but it's been true in many, many other areas of science and technology. And I think it's important to realize that. Third, in my experience, has been exposure to very different disciplines. I think it really stretches you when you get exposed to different kinds of things. For example, again, my own career, I'm a chemical engineer, but I worked in a surgery lab in a hospital, and I was the only engineer there. And I got exposed to things that I didn't even know about, but I could see by talking to the medical doctors who were there that, boy, you could solve different problems using engineering and using chemistry, and that hadn't really been done very much before. So that really helped me. And I found, like in the building I'm in, the Koch Institute at MIT, we have one building, but it has, and cancer is certainly one of the targets, but there's biologists in the building, all kinds of medical doctors, and 7 different types of engineers, like chemical engineers, mechanical engineers, material scientists. I think that helps everybody. Fourth is to take risks, and to realize that failure is okay. But risk taking, goes hand in hand very often with things that make a big impact. But if you take a big risk, you could fail, and that's okay. Fifth is that if your idea is important, I think you can expect lots of criticism. I've certainly had that, and I think everybody who's done, sort of science in a significant way, or invented things, they'll get a lot of criticism, and that's unfortunate, but that's just the way the world works. And that leads me to my last point: whether you get a lot of criticism or anything else, almost never give up. I mean, I probably err on the side of not giving up, but I still think, maybe, after many, many years, or if there's some other, non-scientific reason, maybe you give up. But I would be, if you have created an invention or discovery that's really a platform that can be used for many things, I wouldn't give up. And certainly nanoparticles have been an example of that. People told me it was wrong, it wouldn't work, and sometimes it didn't. Nonetheless, if we didn't have the nanoparticles, you wouldn't have the COVID vaccines. That's been key to Moderna and other companies. So I think it's just really important never to give up as well. Thank you very much.